Hello students, welcome back to the new video from Gokuldham International School. Today we are going to start a new chapter, that is plant growth and development. We all are very familiar with the term called growth and development. In this chapter we are going to discuss about growth and development in a scientific term, and we will also discuss about the certain hormones that is responsible for the uh, growth and development, related to growth and development. So let us discuss about growth and development. First of all, we will discuss about growth. First is introduction. All cell of the plant develops from the zygote. We all know that all the organisms and all the plant cells that we see are developed from the zygote, and the zygote produce a number of cells which organize into tissue and organ. We know that the zygote divide and redivide and then produce a number of cells which later organize into tissues and organs. Development is the sum of two processes. First one is a growth and second one is a differentiation. Means development is the process that is sum of two process. First one is a growth and second one is a differentiation. During process of development, a complex body organization is formed that produce root, leaves, branch and other organisms or other organs, and later this organism eventually die. You can see in this picture that from seed new plants are born, and on that plant many different kinds of organs are produced. Now let us see what is growth. Growth may be defined as irreversible permanent increases in a size, volume or mass of the cell or organs or whole organisms known as growth. In simple terms, we can say that the growth is the permanent and irreversible increases in a size, volume and mass of the cell or particular organism known as growth. Means जो organism है उनमें organism या organs है उनमें जो irreversible permanent change होता है उसे हम growth बोलते हैं. Growth is the one of the fundamental characteristic of living beings. Means all the living things are one similar, having one character, that is growth. Means सारे लिविंग ऑर्गेनिज्म में ग्रोथ एक इम्पोर्टेंट एंड कॉमन कैरेक्टर्स है जो कि जिसमें हर लिविंग ऑर्गेनिज्म में ग्रोथ होता है. That is suggested by this statement that growth is one of the fundamental characteristics of living beings. It accomplished by metabolic processes, for example anabolic and catabolic processes that occur at the expense of energy. Means growth is take place by the acquiring energy. Means they require energy for the growth, and the example of growth is expansion of leaf and elongation of the stem. Now characteristic of growth. The main characteristic of growth are cellular growth, cell division, cell expansion and cellular differentiation. Now let us see next point is growth rate. Growth rate can be defined as increases growth per unit time. Growth rate means the measurement of growth is in terms of increases of growth per unit time. Means कोई पर्टिकुलर टाइम में कितना ग्रोथ होता है जिसे हम बोलेंगे ग्रोथ रेट. द rate of growth can be expressed mathematically, and there is two type of growth rate. First one is arithmetic growth and second one is a geometric growth. Arithmetic growth, let us see what is arithmetic growth and geometric growth. First one is arithmetic growth. After meiotic division, only one daughter cell continue to divide while other differentiate and mature. Means in arithmetic growth, the as a result in a arithmetic growth, the cell that divide and produce two daughter cell Out of two daughter cells, one cell continue to divide and other cell mature, differentiate and mature into a new permanent cell. For example, root elongation at the constant rate. It is mathematically expressed by LT is equal to L0 plus RT. Here LT is length at time and L0 is length at a time 0, and R is the growth rate per elongation per unit time. Now next is geometric growth. The initial growth is slow, which is known as lag phase, followed by the rapid increases in growth, which is known as exponential phase, and followed by the phase where growth slow down, known as stationary phase. The examples of this geometric growth is all cells, tissues and organs show this type of growth. If one plots the parameters of growth against the time, it would be a typical S curve. The exponential growth can be expressed as W1 is equal to W0 ERT, where W1 is final size in terms of weight, height and numbers, and W0 is the initial size at the beginning of the period, and R is the growth rate, and T is the time of growth, and E in the equation is base of natural algorithm. The graph for the geometric growth is shown here, where it plotted in a growth against the time, and it shows the three different phases: lag phase, exponential phase and stationary phase. Now let us see next point is differentiation, dedifferentiation and redifferentiation. So let us see first of all differentiation. Cellular differentiation is the process by which a less specialized cell becomes a more specialized cell. In simple terms, we can say that differentiation is the process in which a less specialized cell converted into a more specialized cell. For example, meristematic cell converted into the parenchyma, xylem, phloem, etc. etc. Means it shows that a meristematic cell converted into permanent tissue and permanent type of tissue or cell. For example, parenchyma cells, sclerenchyma cell, and other cells. Cells derived from the meristematic cell and cambium cell differentiate and mature to perform specific function, which is termed as a differentiation. Means a meristematic tissue that or cell that converted into a mature cell to perform a specific function, which is known this process known as differentiation. Now next is dedifferentiation. Cell undergo structural changes during differentiation. मीन्स जब डिफ्रेंशिएशन होता है तब सेल में कई तरह के डिफरेंट स्ट्रक्चरल चेंजेस होते हैं. चेंजेस टेक प्लेस बोथ इन सेल वॉल एंड प्रोटोप्लाज्म. इट मीन्स जब डिफ्रेंशिएशन होता है तब सेल वॉल एंड प्रोटोप्लाज्म दोनों में स्ट्रक्चरल चेंजेस आते हैं. An example of this is cells lose their protoplasm during formation of tertiary elements. For example is when the examples of differentiation is when tertiary elements are formed. At that time, a meristematic cell lose their protoplasm and converted into tertiary elements, which main function of this is to transport the water. Plant develop a strong elastic and lignocellulosic secondary cell wall to carry water to long distance even under extreme tension. Now let us see what is dedifferentiation. Dedifferentiation is an important biological phenomenon whereby cells regress from the specialized function to similar state of function like stem cell. Means in dedifferentiation, a differentiated cell converted into again meristematic cell, and it gained the ability to redivide. This process is known as de-differentiation. In simple term, we can say that de-differentiation is the process in which plant or a cell converted into again converted into meristematic cell and regain the uh, cell division capability. An undividable differentiator cell sometimes regain the power of division, and this process is known as de-differentiation. Means जो मैच्योर सेल है वो अगेन सेल डिवीजन की कैपेसिटी गेन करते हैं इस प्रोसेस को, और सेल डिवाइड होते हैं, और इस प्रोसेस को हम लोग डी डिफ्रेंशिएशन बोलते हैं। जो मीन्स जो डिफ्रेंशिएशन हो चुका है उसमें वो कन्वर्ट होगा, जो सेल डिफ्रेंशिएट हो चुका है वो मेरिस्टेम में अगेन दोबारा कन्वर्ट होगा, उसे हम बोलेंगे डी डिफ्रेंशिएशन। मीन्स इट इज a reverse of differentiation. A de-differentiation is a common process in plant during secondary growth and wood healing mechanism. मीन्स जब सेकेंडरी ग्रोथ होता है तब परमानेंट टिश्यू डी डिफ्रेंशिएट होके मेरिस्टेमिक टिश्यू में कन्वर्ट होते हैं, और वो मेरिस्टेमिक टिश्यू न्यू सेल्स को प्रोड्यूस करते हैं, जो जिसे हम सेकेंडरी ग्रोथ बोलते हैं। और डी डिफ्रेंशिएशन इज वेरी इंपॉर्टेंट प्रोसेस इन टर्म्स ऑफ secondary growth in plants. Now let us see what is redifferentiation. A redifferentiation is the process in which the de-differentiated cell again converted into mature cell, which is known as redifferentiation. So let us see what is redifferentiation. A de-differentiated cell can divide and produce new cell. A new cell produced again lose the power of division and become a part of permanent tissue, which is known as redifferentiation. It means जो डी डिफ्रेंशिएटेड सेल है वो अगेन न्यू सेल्स प्रोड्यूस करेंगे, और ये न्यू सेल अगेन सेल डिवीजन की कैपेबिलिटी लॉस कर देंगे, जिसकी वजह से वो अगेन वो सेल डिवीजन नहीं कर सकते, एंड देन इट कन्वर्टेड इनटू परमानेंट सेल और टिश्यू, which is known as redifferentiation. In simple term, we can define this redifferentiation as a redifferentiation is the process in which a differentiated cell produce new cell and it lost the capacity to divide and converted into a mature cell again, and this process is known as redifferentiation. The example is formation of tumor cell, which is known as redifferentiation, which is formed by the process called redifferentiation. Now this is about growth, and now let us see about the development. So what is development? As I already mentioned, as development is the sum of the growth and differentiation. मीन्स जो डेवलपमेंट है वो दो प्रोसेस से बना होता है जिसमें ग्रोथ एंड डिफ्रेंशिएशन इन्वॉल्व होते हैं, और वो ग्रोथ एंड डिफ्रेंशिएशन का सम होता है डेवलपमेंट. नाउ लेट अस सी व्हाट इज डेवलपमेंट. डेवलपमेंट is a term that include all the changes that an organism goes through during its life cycle from germination of seed to senescence. मीन्स उसकी लाइफ में जब सीड में से वो जर्मिनेट होता है तब से लेके और सीनेसेंस तक के दौर को हम डेवलपमेंट बोलते हैं. Here in the figure you can see that first meristematic cell divide and redivide and produce plastic growth, then it differentiate and become mature, and mature cell later senescence or it death. It means the mature cell go into the senescence or dying. So all the process is known as development. The sequence of developmental process in plant. This is the figures of the sequence that take place in the plant cell. Now next point is plasticity and heterophiling. The plasticity, the ability of plant to follow the different pathways and produce different structure in the response to environment and a phase of life. For example, in cotton and coriander, the plant, the leaves of the juvenile plants different in the shape from the those mature plant. Means जो plants कॉटन एंड कोरिएंडर प्लांट होते हैं उसमें जुवेनल लिव्स एंड मैच्योर लिव्स डिफरेंट डिफरेंट होते हैं, डिफरेंट फॉर्म के होते हैं, जो कि एनवायरमेंटल कंडीशन एंड डिफरेंट पाथवेज की वजह से होते हैं और डिफरेंट फेजेस ऑफ लाइफ की वजह से होते हैं, जिसे हम प्लास्टिसिटी बोलते हैं. एंड देन नेक्स्ट टर्म इज heterophyling. The phenomenon of appearance of different form of leaves on the same plant is known as heterophyling. मीन्स एक ही प्लांट पे दो तरह डिफरेंट तरह के लिव्स देखने को मिलते हैं, जिसे हम heterophyly बोलते हैं. द एग्जांपल्स ऑफ heterophyly इज अकर इन कॉटन एंड कोरिएंडर. Now next topic is plant growth regulators. Plant growth regulators, which is also known as phytohormones. And what is now let us see what is this phytohormones or plant growth regulators. So the phytohormones or plant growth regulators are chemical substances that influence the growth. Means plant growth hormones are the chemicals that influence the growth. मीन्स वो ग्रोथ को इन्फ्लुएंस करते हैं. These plant growth regulators function as a chemical messenger for the intercellular communication. Means these plant growth regulators function as a messenger for the intercellular communication. They work together coordinating the growth and development of the cell. Means they coordinate and develop together and control the growth and development of the cell. They could be indole compounds or adenine derivatives, and they may be gases. And let's see the different forms and different types of plant growth regulators. There are mainly 5 different phytohormones. First one is the auxin, second one is the gibberellin, third one is the cytokinin, fourth one is the abscisic acid, and last one is the ethylene. The classification of plant growth regulators are the plant growth regulators divided into 2 groups. First one is the plant promoters and growth promoters and growth inhibitors. The name itself suggests everything that plant promote. Growth promoters means they promote the growth, and growth inhibitors means they inhibit the growth of the plant. So in a growth promoter, 3 phytohormones are included. First one is the auxin, gibberellin and cytokinin. And in growth inhibitors, 2 different phytohormones are involved. First one is the abscisic acid and second one is the ethylene. Now let us see first phytohormones, auxin. Auxin was discovered by Charles Darwin and Francis Darwin. F.W. Wentz isolated auxin from the tip of coleoptile of the oat seedling. Wentz developed the Havana culture test for the bioassay of the auxin. The different types of auxins and examples of auxins are here. First one is the IAA, which is stand for indole acetic acid, and then IBA, indole butyric acid, then NAA, naphthalene acetic acid, and then 2,4-dichlorophenolic. And from these different types of auxin, indole acetic acid or IAA is the only natural auxin found in the plants or naturally, and others are synthetic auxins which is used for in agriculture nowadays. Now let us see the physiological effect of auxins, means how this auxin are effect on the plant. फर्स्ट वन इज दे प्रमोट द एपिकल डोमिनेंस मीन्स जो एपिकल होती है एपिकल टिप ऑफ द प्लांट दे प्रमोट द एपिकल ग्रोथ ऑफ द प्लांट एंड सप्रेस द लेटरल ग्रोथ. It initiate the rooting in the stem cutting means it promote the advantageous rooting in the plant. It promote the flowering in a pineapple means वो ऑक्सीजन होता है जो पाइनएप्पल में और कई सारे प्लांट्स में अब फ्लावरिंग को प्रमोट करता है. देन इट प्रिवेंट द फ्रूट एंड लिप ड्रॉप एट अर्ली स्टेजेस मीन्स जो ऑक्सीजन होता है वो प्लांट में जब अर्ली स्टेजेस में लिप फॉल एंड फ्रूट फॉल होता है उसे वो अरेस्ट करता है मीन्स प्रिवेंट करता है. देन इट प्रमोट द एब्सेशन ऑफ ओल्डर मैच्योर लीव्स एंड फ्रूट मीन्स ऑक्सीजन होता है वो एब्सेशन करने में हेल्प होता है जो कि ओल्डर टिश्यूज को एब्सेशन और सिनेशन में हेल्प करता है. इट इंड्यूस द पार्थेनोकार्पी इन टोमेटो मीन्स पार्थेनोकार्पी मीन्स अ फॉर्मेशन ऑफ फ्रूट विदाउट अ सीड एंड दिस ऑक्सीजन promote the promote or induce the parthenocarpy in the fruits. Examples is tomato. It is also used as a herbicide and control the xylem differentiation and help in the cell division. Now next is gibberellin. The gibberellins is a group of another phytohormones that is discovered by the E. Kurosawa, a Japanese scientist, has identified the gibberellins present in fungal pathogen Gibberella fujicorum. मीन्स ये जो जापानीज साइंटिस्ट है उन्होंने फंगल पैथोजन जो कि जिब्रेला फिजिकोरी है उसमें से जिब्रेलिन को डिस्कवर किया था. Gibberellins were isolated from infected dry seedling when treated with the sterile filtrate of fungus. And the physiological effects of gibberellins are the elongation of intake stem means it is very useful and it promotes the internodal elongation in plants like sugarcane. Then drop shoots, it also used in drop shoots to promote the internodal elongation. Then molting in rosette plants like cabbage and hinabin and overcome the natural dormancy of blood, tumors and seed. Gibberellins are very useful in the seed dormancy and it helps in the seed germination. It induces the parthenocarpy or development of seedless fruit, which I also mentioned in oxygen that this type of plant growth hormones are useful for the inducing of parthenocarpy and development of seedless fruit. And it promotes the flowering in long day plants means ये long day plant में flowering को introduce करते हैं और induce करते हैं. Then it is used as a vernalization. Now let us see the next phytohormones. Next one is cytokinins. Skoog and Miller discovered the cytokinins. Skoog and Miller crystallized the cytokinesis promoting active substance name is kinetin from corn kernels and coconut mills. Means Skoog and Miller who discovered the cytokinins or chemical substance which promote the cytokinesis during the cell division, and they named it is a kinetin and later it is converted into cytokinins or later it is known as a cytokinin. Now let us say the physiological effects of cytokinins. The main and important effect of the cytokinins is they induce the cell division. It means they increases the cell division in plants. Then they delay the senescence of leaf and other organs. Means we can we can arrest the senescence in leaves and other organs by applying the cytokinins on the plant. They promote the lateral bud, means they promote the branching of the lateral bud or development of the lateral buds. They increases the cell expansion in dicotyledons and in leaves, means ये जो साइटोकाइनिन है वो सेल एक्सपांसन एंड इन डाइकोट प्लांट में जो डाइकोटाइलिडन होता है जो कि लीव्स होता है उसको एक्सपांसन करने में हेल्पफुल होता है. And then it promote the chloroplast development and chlorophyll synthesis, means by applying cytokinin we can promote the chlorophyll production and development of chloroplast in the plant. So these are the physiological effect of the cytokinins, means this is the cytokinin and auxin are two main uh, hormones or phytohormones that is used in tissue culture to promote the different differentiation and to produce leaves and shoot and roots in the plant in the callus in the tissue culture. Now next is these three are auxin, gibberellins and cytokinins are plant growth promoters. And now let's see the plant growth inhibitors. First one is ethylene. Auxin confirm the release of volatile substance from ripened organs that hastened and ripening of stored and unripe bananas. The volatile substance was identified as the ethylene, means certain these scientists who discovered the volatile substance that induced the ripening in non-ripening fruit, and later it was founded or identified that it is a ethylene, which is also known as a ripening phytohormone, which means it induces the ripening in different fruits and different organs. Now let us see the physiological effect of ethylene. Ethylene promotes the senescence and abscission. It inhibits the elongation of stem and root, means it promotes the senescence or death of the cell or tissue, and then it inhibits the growth of stem and root or elongation of the stem and root. Then it induces the fruit ripening. That's why it is known as fruit ripening hormones. It breaks the seed and bud dormancy, and it initiates the germination in peanut seed, means it help in the germination of peanut seeds. Then spoilating of potato tubers, means it is helpful in the spoilating of tomato tubers. Then it promotes the root growth and root hair formation, मीन्स जो इथेलिन होता है वो रूट के ग्रोथ को प्रमोट करता है एंड जिसकी वजह से वो रूट हेयर फॉर्मेशन में हेल्पफुल होता है. देन इट इनिशिएट द फ्लावरिंग एंड फॉर सिंक्रोनाइजिंग द फ्रूट सेट इन पाइनएप्पल्स, means it is helpful in the flowering and for the synchronizing of fruit set in the pineapple. So these are the different physiological effects of ethylene. Now let us see the next one is a next phytohormone is a abscisic acid. Abscisic acid is a one of the growth inhibitors, and this abscisic acid, which is also known as ABA, in 1963 Friedrich T. Endicott and his co-worker identified the abscisic acid. Abscisic acid was isolated from several abscission or accelerating substance from the cotton plant, means जो ये abscisic acid है वो abscission accelerating substance कॉटन में से आइसोलेट किए गए हैं एंड इट इज नोन एज स्ट्रेस हार्मोन. दिस कैन बी आस्क्ड इन एग्जाम दैट एब्सिसिक एसिड इज आल्सो नोन एज, सो द आंसर इज स्ट्रेस हार्मोन. द फिजियोलॉजिकल इफेक्ट ऑफ साइटोकाइनिन इज or are the the abscisic acids are act as a general plant growth inhibitors, which means it act as a plant growth inhibitors. It induce the seed and bud dormancy, which means it induce the seed and bud dormancy, and it inhibit the seed germination, means जो ये एब्सिसिक एसिड है वो सीड जर्मिनेशन को इनहिबिट करता है। मीन्स अगर सीड में ज़्यादा मात्रा में और ज़्यादा परसेंटेज में एब्सिसिक एसिड है तो वो सीड जर्मिनेट नहीं हो पाएगा, एंड देन इट स्टिमुलेट द क्लोजर ऑफ स्टोमेटा। मीन्स इफ द एब्सिसिक एसिड द concentration of एब्सिसिक एसिड is increases, then stomata will close, and this abscisic acid play an important role in seed development and maturation and dormancy. Means this abscisic acid is a phytohormone which is also known as stress hormones. It play an important role in seed development, maturation and in dormancy. Now let us see next point is a photoperiodism, which is an important phenomenon that take place in plants. The photoperiodism. So let us see what is the photoperiodism. Photoperiodism is a response of plant to relative length of the light and dark period. Means ये photoperiodism है वो एक तरह का रिस्पॉन्स है ऑफ प्लांट, विच मीन्स it is dependent on the length of light and dark period. Means the photoperiodism is the response of plant to relative length and the darkness of the darkness period. Means ये जो फोटोपीरियडिज्म है वो उसकी वजह से प्लांट रिस्पॉन्स करता है कि अगर लेंथ ऑफ द लाइट इज on the basis of length of light, plant response, which is known as plant photoperiodism. Some plants require periodic exposure to the light to induce flowering, which means so there are certain plants which require high length of the light or longer daylight so for the flowering, and duration of the dark period is equally important for the flowering. मीन्स द जो डार्कर पीरियड होता है वो भी फ्लावरिंग के लिए इम्पोर्टेंट होता है ऑन द बेसिस ऑफ द लेंथ ऑफ द लाइट और लेंथ ऑफ द डे लाइट required by for the flowering in different plants. It is divided, the plants groups are divided in the three groups. First one is a long day plant, short day plant and day neutral plants. So let us see, first one is a long day plant. Plants that require exposure to the light for period and more than critical duration to induce the flowering, which is known as long day plant. The examples is pea, barley and wheat. Now short day plant that require exposure to the light for a period less than critical period to induce the flowering, which is known as short day plant. And examples of short day plants are rice, jawar and cotton. Means short day plants are plants that require short day length and it require long dark period, so it is known as short day plants. And these short day plants are grow in a winter season, where long day plants are grow in a summer season. Now day natural plants where there is no correlation between the exposure to light duration and induction of flowering. Means उसे day neutral plants are those plants there is in which there is no correlation between exposure of light and induction of the flowering. The examples of these plants are cucumber, rose and tomato. Now next is vernalization. So what is vernalization? Vernalization is the phenomenon of induction of the flowering in plant by exposure to the lower temperature. Means Vernalization is the process or phenomenon to induce the induction of the flowering. मीन्स फ्लावरिंग जल्दी प्रोडक्शन करने के लिए हम लोग सीड्स को प्लांट्स को अर्ली स्टेजेस में हम लो टेम्परेचर पर रखते हैं, जिसकी वजह से उसमें वर्नालाइजेशन होता है एंड उसके जिसे हम वर्नालाइजेशन बोलते हैं, जिसकी वजह से अब फ्लावरिंग जल्दी होगी उसकी लाइफ में और प्लांट की लाइफ में. It prevents the precocious reproductive development and late in the growing season and enable the plant to have sufficient time to reach the maturity. Example is biennial plants. These are the monocarpic plants that flowers and then die in a second season. Examples is sugar beet, cabbage and carrot. So vernalization, so in short term, in a simple term, we can say that vernalization is the process or phenomenon in which we induce the flowering in a plant by exposure them to the low temperature. मीन्स प्लांट्स को लो टेम्परेचर पे रखने की वजह से उसमें जल्दी से फ्लावरिंग होगी, उसे हम बोलेंगे वर्नालाइजेशन. एंड वी डज इट इन अ बायनियल प्लांट्स एंड these are the monocarpic plants that flower and then die in a second season. The examples is carrot, sugar beet and cabbage. So here in this chapter we discuss about growth, development and certain plant growth regulators. Plant growth regulators are important for these board exams. And in plant growth regulators we discuss about two different types of plant growth regulators. First one is the growth promoters and growth inhibitors. In growth promoters there is three different phytohormones. First one is the auxin, cytokinin and gibberellin. And in growth inhibitors there is two different phytohormones. First one is ethylene and abscisic acid. And then we discuss about photoperiodism and vernalization. So here we completed our chapter growth and development with the piece. Thank you students.